Okay, we'll get started for today. In today's session, we'll discuss mainly about constant-based modeling, variations of those, including flux-based analysis, flux balance analysis, and some other variations like basically MOMA or ROM. We'll talk about those in the later part of the session. And at the end, we'll solve some MCQs like we do. So let's get started. Okay. So the first concept required to understand how metabolic modeling works and how these models are created and analyzed. So first important thing to know is the stoichiometric matrix. So what is it? It is basically a matrix of the coefficients which are proportional to the substrate and the product molecules involved in the reaction. Say for example, you have a reaction like something like this, A plus B giving 2C. So the stoichiometric coefficients for A, B, and C are generally minus 1, and 2 respectively, because this minus sign represents that the uh, substrate is getting used up in the reaction, and the plus sign for the product uh, indicates that the products are getting formed during the reaction. So the coefficients for the uh, elements involved in the reaction are minus 1, and plus 2. And now let us assume that our metabolic network has the reactions that consists of the reactions R1 to RR. So we have R number of reactions inside the metabolic network, and each such reaction is denoted by the stoichiometric coefficient of those particular reactions. Now, let us assume that the reaction Ri operates with rate Vi, and we can collect these individual reaction rates to a rate vector. So uh, this is basically a column matrix consisting of all the rates of the reactions of all the reactions in the metabolic network. So you have R reactions. So you'll have rate R rate of those reactions as a vector, as a column vector, which is denoted by this V vector here. Now, similarly, the concentration vector will be a column vector consisting of all the concentrations of each metabolite in that system at time t. We'll see how these are represented. Now, the stoichiometric coefficient of the reactions are collected in a vector called SR. So basically, SR looks something like this. So there is one position for each metabolite in the metabolic system, and the stoichiometric coefficient of the reactions are inserted in appropriate positions. So for this reaction, A plus B given 2C, your SR vector, that is the stoichiometric collection of the stoichiometric coefficient of the reactions, are in this SR vector. Now, these SR vectors are basically added, not added, you can use the term concatenated, and they can be combined into the stoichiometric matrix S. Now, what is the representation in the matrix? So, yeah, this is your matrix. There's one row for each metabolite. So basically, M1 uh, uh, to MM are the number of metabolites here. So there are basically M metabolites, and one each of the column represents each reaction. So your matrix of the form M cross N will have M metabolites and N reactions, and the coefficients of these of the basically the jth column along the coefficients of Sj along the jth column are the stoichiometric coefficients of the reaction J. So basically these are representing each of your reaction, these columns. Now the coefficients of these of the vectors present in these columns represent the stoichiometric coefficients of the whole reaction J. Okay. Now we have seen how a stoichiometric matrix looks, and what are the different meanings, or like, what are the row and the column represents. Now let us look at something called steady state approximation. So the systems equation, the systems of equations that you have. So you have a basically this is your stoichiometric coefficient matrix. You have another matrix consisting of all the column, basically a column vector of all the reaction rates of each of those reactions. So that is given by this vector v. So as a system of equations, this can be represented as dA of xi dt is basically summation of all these or summation of the multiplication of all these individual elements along with the column vector. So basically, this is basically matrix multiplication. If you imagine a column vector of say n elements, and your matrix is basically n cross n, so you're multiplying a matrix of n cross n with a matrix of n cross 1. So your basically your uh, the react basically the answer that you get, the result that you get will basically n cross 1. So this SI matrix, this is representing this matrix. So the SI matrix contains all the stoichiometric coefficient of the single metabolite, that is the row of the stoichiometric matrix, and the system of different equations, differential equations. This is a typo here. And the system of differential equations together can be then be expressed by the matrix equation, which is basically dx dt equal to SV. Note that this transpose is gone here because the dx dt vector is already like a transpose of the original dx dt vectors. So this transpose, you do not need to do another transpose here. So yeah, basically it collects the rate of all this dx dt. What it is doing is basically collecting the rates of concentration concentration changes of all metabolites in the system under in the metabolic platform. Now, most applications of this stoichiometric matrix assume that the system is in so-called steady state. So basically, when we have a certain set of reactions and the reaction rates, and basically you have a set of reactions with the coefficients and you have a set of reaction rates, you want to find out basically the reaction rates are unknown, so you want to find out the reaction rates from the from dxdt and s. So uh, the natural way to do it is to assume steady state approximation. So in steady state, the concentrations of these metabolites remain constant over time. Therefore, this dxdt, dxidt, that is representing each basically metabolite, the rate of change of this metabolite is zero in steady state, and which is basically meaning that the production is equal to the consumption of each metabolite, which forces the reaction rate to be invariant. So one small application here is that basically you are equating your S into your V vector equal to 0. So basically your V vector, the vector representing all your reaction rates, is in the null space of your S matrix. Remember we saw a little bit of matrix algebra earlier. Just a second. So yeah, so basically, yeah, you're equating your S dot V equal to 0. So basically, your V is the vector containing all your reaction rates. That is basically present in the null space of the S matrix. So this is a small terminology that you may see uh, across different literature as well. They're mentioning ki, like when you're trying to find the reaction rates, the vector of the reaction rates is it's generally in the null space of the S matrix. This is what it means. Basically, in steady state, you are equating the product of your stoichiometric matrix and your velocity and your basically rate vector equal to 0. So your rate vector is in the null space of your stoichiometric matrix. So this is what basically it means. Okay. Next, we come to the understanding of metabolic network dynamics. So first is the representation. Metabolic reactions are depicted using stoichiometric matrix that we just discussed, sized n cross n. So each row signifies unique compounds or metabolites, and each column represents each of the reactions in the metabolic network. Entries in each column are of the stoichiometric matrix determine the stoichiometric coefficient of the participating metabolites, like we saw here. So basically, these are all the stoichiometric coefficients of all the metabolites in the reaction, and uh, it is basically negative for consumption and positive for production, and zero for non-participation. So uh, just to give you a little bit of heads up, these stoichiometric coefficients are are also used as constraints for your model. So, for example, in your model, you want to emphasize on the fact that a particular metabolite is not participating, or it is not participating in the metabolic network after getting, say, for mutated or deleted or something. So you have a metabolic network, you're checking how the flux will change in the network when you change the uh, when you basically delete a gene or delete a protein. So in those kind of cases, you would like to constrain your set of solutions to the point that that metabolite should have a zero as a stoichiometric coefficient after basically after getting the results of the model. So these kind of uh, this kind of values are basically put as constraints to your model to basically push the model to a specific outcome. Okay. Now, next comes the concept of flux and concentration. So, flux is basically the typical meaning of flux is basically the rate of flow of something. So, we have learned electric flux in our in our school days, like in 11th, we learned something called electric flux. Here, we are learning something called metabolic flux. So, how much of energy or how much of metabolic elements are flowing from one end of the network to another end of the network. This is something you can imagine as flux. Now, these fluxes are represented by this vector. So the flux is representing the reaction rates here in this case, but it can represent other things as well. So in this case, we are assuming the flux to be the reaction rates, but in other cases, in other complex cases, the reaction rates may not be the flux. So there, you need to be a little bit careful about how you're defining flux. So you're here, we are assuming that the vector v, which are all the reaction rates for all the metabolic reactions, is representing the flux, and the concentration of the metabolites are depicted in the vector x, the one basically this vector x, the one we are equating to zero. So dx dt is the rate of change of the concentration of each metabolite. Next comes the mass balance equations. So at steady state, as we define, the rate of change of the uh, metabolite will be zero, and the system can be expressed as SV equal to zero, and your V will be the null space in the null space of F. And any V satisfying this equation resides in the null space of S. But there are some challenges when you use these kind of network dynamics to model a particular metabolic network. So what happens is, in real life cases, realistic metabolic models will have more reactions than compounds. So number of compounds in the cell is the limiting factor basically in metabolic reactions. So the number of metabolites will be much lower than the number of reactions. One reason for that will be a single metabolite will be involved in a uh, number of metabolic reactions. So for example, in glycolysis and Krebs cycle and TCAC, we have these are basically metabolic reactions that are happening, but they'll have some common elements. So glycolysis will have some common elements of Krebs cycle and Krebs. Basically, the end of glycolysis will be the start of Krebs cycle. So the end of the glycolysis, the acetyl CoA basically formed by at the end of the glycolysis, that will be taken up as a metabolite in the Krebs cycle. So two of two different metabolic networks are sharing a single metabolite. So there are numerous examples of these kind inside the cell. So this is a very realistic issue that the number of reactions will be much more than the number of uh, metabolic compound. So your number of columns will be much more than the number of rows. So it will be kind of a fat matrix. So what it the problem it causes is basically it will lead to an undetermined system where the number of unknowns will exceed the number of equations, and hence it will lack a unique solution. So yeah, this is a challenge that is faced in real life metabolic models. Now, identifying optimal solutions from these kind of this kind of set of equations that all satisfy your constraints. So basically, you do not have a unique solution; you have a set of solutions, and you have to identify the optimal set of solutions from those your original set. Now, uh, despite constraints defining solution ranges, pinpointing specific points within the solution space is feasible. So it is possible like you can draw out the whole curve along which your constraint is acting, and you can pinpoint specific places in the curve. Say, for example, a local minima you're seeing in the surface. You can pinpoint to the local minima and claim it to be one of the most feasible solutions of your system of equations. So there are like if you have constraints on the set of solutions that you are going to get, so you can pinpoint an area of the in the 3D space or in the n-dimensional space, depending on how many reactions and metabolites are involved. So in the dimensional place, n-dimensional space, there will be a curve, a part of the curve that will signify all the solutions that are following your constraints of the model. So that is possible. Now, examples include identifying maximum growth rate or ATP production under organism-specific constraints. So, we will look at one of the examples of this, and one of the methods that basically follow these kind of this set of uh, this set of steps to model a metabolic network is flux balance analysis. So we'll look into it what it is. It is a method for pinpointing optimal points within a constraint within constraint spaces. So you have constraint space and you want to pinpoint certain most optimal points in that's your solution space. So that can be done using flux space analysis, flux panel analysis, and we'll look at it how it is done. Okay. So before we move on to flux balance analysis, so flux panel balance analysis is a part. It is a method that comes under constraint based model. So first we'll learn about what constraint based modeling is, and then we'll learn about what flux balance analysis is. So constraint-based modeling, it is an approach based on the assumption that organisms exist in particular environment that typically have scarce resource, and over time, the probability that the fit will survive is higher compared to the probability of the survival of the less fit. Basically, our evolutionary Darwinian principle. Uh, Remember, we saw these kind of principles applied to genetic algorithms in the previous classes. This is one such example where evolutionary concepts can be applied to constraint-based modeling. So, to be fit or for survival, a myriad of constraints must be satisfied, which limits the range of available phenotypes. So, uh, you will have a set of solutions. You will generally have a fat matrix denoting metabolic, co- co- met- basically the metabolic elements involved in the reaction, and you'll have a number of solutions. You'll have not have a unique solution. You will have a number of solutions denoting a space in the n-dimensional space, denoting a curve in the n-dimensional space. Now, uh, uh, on the curve, on your solution set, you'll have some constraints which must be satisfied, and the constraints will be in line with the uh, Darwinian evolutionary theory that uh, nature selects all. Uh, Okay. Yeah. Uh, yeah. On your solution space, there will be constraints based on Darwinian's evolutionary principle that the fittest will survive. What is fittest and what is survival? We talked a little bit about these concepts in genetic algorithms, but similar concepts can be applied here as well. So, the constraints that you are applying uh, on this solution space to get your optimal solutions, the specific optimal solutions. There can be different categories of these constraints. First can be physiochemical constraints. So these are like mass balance, energy balance, momentum balance, these basic fundamental laws of nature. Basically, these uh, conservation of energy principles that we have. So those can be used as constraints. Furthermore, biochemical reactions must always result in a negative free energy change to proceed in a forward direction. So we call a little bit about thermodynamics studied in your plus two. So basically, your if your free energy is negative, that means your products are at a lower energy level than your substrates. So this is a condition that is very often, not often, like in case of 99.9 percent of the reactions that we see in biochemical systems, this is a feature that is conserved across all. So this is a constraint that you can apply in your model to get an optimal set of solutions. Another type of constraint will be topobiological constraint. So the crowding of molecules inside cells will lead to these kind of constraints. So the concept of static hindrance that we learned in our plus twos as well. So you cannot accommodate all those molecules together in one place because the size of the cell or the size of the compartment of the cell where the reaction is occurring, that is a limiting factor. So you cannot have more molecules than what you can fit inside that space. So those kind of constraints can also be applied. There can be environmental constraints as well, nutrient availability, pH, temperature, etc., etc. So those can be limiting factors as well. And also there are something called regulatory constraints. So flux balance analysis doesn't take into account these regulatory constraints. We'll look at a different method for that. But these are also constraints, uh, but these are not basically hard constraints that we saw in the earlier, basically the other types of constraints, the physiochemical constraints or the topobiological constraints, etc. So these regulatory constraints, they are not hard constraints, but they are restraints. So they are implemented in the cell in various ways, including amount of gene products made and their activity. So, for example, uh, a gene product a gene product forming that is kind of forming a feedback loop, a feedback auto-regulatory loop, which is basically uh, like forming more of itself. It's kind of a fractal. So you have a product which is going and sitting as a transcription factor in that particular gene only, and it is forming more of the products. Now, this is a condition if like if this system is allowed to go on for infinite amount of time, so this will grow and grow and grow. There will be no saturation. But as we see in biochemistry, there's always a level of saturation that is reached after which the products will not be formed. So there are regulatory mechanisms on these genes and gene products and this protein formation about how much protein or how much expression of a particular gene is required for the cell to function, or how much of that concentration of the particular metabolite is required for the cell to survive. So there are certain constraints on that on the side of the cell as well. So cell will not make products that it will not use in the future. So the energy, the cell will also always try to minimize its energy consumption. So it will not make unnecessary gene products that it will ultimately have to degrade again. So there will be constraints on the concentration of the gene that is getting expressed, and also also the amount of protein that will be getting formed. So there can be transcriptional and translation regulations. So transcription factors going and binding to gene promoters and regulating how that gene is getting expressed. If there can be other regulatory elements as well, enhancers, repressors, which can control the expression of those genes, and also there is activity in terms of enzyme regulation that can also be controlled. So these regulatory constraints, these are like a little bit trickier to define in comparison to the other type of constraints, the physiochemical or the environmental, etc. These constraints are a little bit harder to define because these are not like hard and fast constraints. Your solution may go out of these constraint boundaries as well. But the point is, uh, those can be termed as those can be termed as exceptional events that can happen in a mutated or a gene knocked out case. But in general, your if your cell is functioning properly, if you have all the genes that are required for it to function properly, there will be certain amount of regulatory mechanisms that constrains the amount of any kind of product getting formed in the cell. Okay. Now, how do you mathematically represent these constraints? Once you put this in this mathematical form, then only you can perform this analysis. So there can be two types. Fundamentally, there can be two types of constraints. So balances, these are constraints that are associated with conserved quantities such as energy, mass, redox potential, momentum, etc., etc. And there can be bounds as well. So these are the constraints that limit numerical ranges of individual values and parameters, such as concentration and fluxes and kinetic constraints. So, majorly two types of fundamental constraints you can represent mathematically. One is a balance. Those are the like conservation of energy equations that you put, and also there can be constraints on the bounds. There can be constraints on the individual range of values that a particular variable or a particular parameter can take, which can include concentration or fluxes, etc. As I said, the conservation of mass is an example of these balance constraint. Mass balance is utilized in terms of flux and stoichiometry of each reaction. Let S be the stoichiometric matrix containing the stoichiometry of all the reactions in the network. We saw how S looks. S is the M cross N matrix where M corresponds to the number of metabolites and N is the number of reactions or fluxes taking place within the network. Uh, the entry as Ij defines the stoichiometry of the reactant I in the reaction as we saw. Now we come to flux balance analysis. Now, so the whole idea here is to balance out the fluxes. So basically, this is also you can think of it as a conservation of energy principle as well. So the flux going into the network, it should be equal to the flux coming out of the network. So the network you can define accordingly. You can have your boundaries accordingly, but in general, the flux should be constant. So here, in another terms, you can also think of it as the constraint of law of mass action. So the amount of metabolite that is moving from one form to another, the the mass of the metabolite moving from one form to another, that must be constant. So how do you do this analysis? So first, you build a metabolic network consisting of a list of stoichiometrically balanced biochemical equations. So this is the network. You curate it using knowledge from literature and other sources. And now, after you've got those reactions and the network, then you convert it into a mathematical model. So you form the stoichiometric matrix, and then at steady state, the flux through each reaction is given by the equation SV equal to 0. As we saw, like you are at steady state, you are assuming that the total that S into V will be 0. Now, since there are more reactions than metabolites in large models, there is more than one possible solution to this equation. And also along with that, an objective function is defined on this. Uh, how is the objective function mathematically defined? It is basically C transpose V, where C is the vector of weights indicating how much each reaction contributes to the objective function. So it is basically a vector of weights of each of the reaction and how much it is contributing to the contributing to the objective function. That is your weights matrix. And you take a transpose of the matrix and multiply it with the reaction rate vector, you get what is known as the objective function. In practice, only one reaction is desired for maximization or minimization, and C is a vector of zeros with a 1 at the position of the reaction of interest. When simulating growth, the objective function will have a 1 at the position of the biomass reaction. We'll look into these examples in next classes, like we will do some like real life examples on how these models are created. There it will be a little bit more clear about how we move about these objective functions. But yeah, this is the whole idea. So you have your metabolic reactions, you form your stoichiometric matrix, you apply your mass balance constraints at steady state, and you define an objective function, and you optimize this objective function using LPP. So linear programming can be used to identify a particular flux distribution that maximizes or minimizes this objective function depending on your constraints and depending upon your model, while observing the constraints imposed by the mass balance equations and the reaction bounds. So this is the whole overview of flux balance analysis. Now let's just talk about how we can like reconstruct this network and analyze basically. So you have to get your data from somewhere. So that can be some literature, some online databases, or some books, etc. So uh, a stoichiometric network reconstruction is assembled piece by piece with gene-protein reactions relationships assimilated from experimental data available in public literature and online databases. And after that, so this you get their data from here and you generate a metabolic reaction network. So this is will be a biochemical reaction. This is in terms of the reactions and metabolites, and this is the stoichiometric matrix. Now, after you've got this, you use FBA to predict a steady state flux distribution throughout the network given certain underlying physiochemical and thermodynamic constraints. And this prediction can be validated with independent experiments. So you have your model, basically you have your stoichiometric matrix, and you do FBA on this, putting S equal to 0, and then you get basically a steady state flux distribution throughout the network under some constraints, and you can validate these results with the help of certain certain experimental techniques. And inconsistencies in this, like differences in results in your experimental data and your predicted data using FBA, those differences can be taken into account and be used for model refinement. And in addition, with the stoichiometric network reconstruction and FBA, the network can be perturbed in different ways as well. So we'll also look at some examples on how the network can be perturbed in the next class. But in general, like when you have a metabolic network, you would want to know, okay, if I knock out a certain metabolite, how is my flux changing, or how is my particular pathway working, or is it not working, or is it getting affected? What is happening? So this kind of questions you'll want to answer with your metabolic network model. And uh, basically you would want to answer how different perturbations can affect different concept different basically production and degradation of certain metabolites. So these perturbations are not basically there can be other ways to assess these perturbations as well. We'll look at one or two examples today as well. So yeah uh, in addition, with the stoichiometric network reconstruction and FPA, the network can be perturbed in different ways, ranging from differences in environmental stimuli to mutations in network structure, whatever be the perturbation. Um, One such example can be single or double gene knockouts, and they can be used to assess the properties of the system, such as robustness, meaning How sensitive is your system? Is your system, if one metabolite is perturbed, is your whole system affected? Or if one metabolite is perturbed, it bypasses that and it tries to minimize the change in the flux throughout the whole network. So these kind of questions uh, you would want to answer from your model. So FPA and its variants are a good way to do it. So yeah, this is one example from that. So and this is basically a classical flux balance analysis approach. So this is a glycolytic pathway on the left side of the figure. This is a glycolytic pathway representing a biochemical network. And the reactions that convert glucose into pyruvate and ultimately into acetyl-CoA uh, in the Krebs cycle basically we've all studied these reactions. So glucose to glucose 6 phosphate to fructose 6 phosphate. So each of these reactions, glucose to glucose 6 phosphate, this is reaction 1. Glucose 6 phosphate to fructose 6 phosphate, this is reaction 2. So V1, V2 up till total we have 12 reactions here. So up till V12 are the different reaction rates given by the V vector. And here in the panel B, you see this is one small example given here. So fructose 6 phosphate uh, converting to fructose 1,6 bisphosphate, biphosphate, and this is uh, uh, how the stoichiometric matrix will look like. So this is basically the R3. This is the third reaction here. So for R3, you have these are the metabolites F6P, FDP, ATP, ADP, and H plus. So basically, ATP is getting converted to ADP, and one H plus is getting released, and fructose 6 phosphate is getting converted to fructose 1,6 bisphosphate or fructose diphosphate here in this case. So this is getting formed, so this is plus one. This is getting degraded, so this is minus one. ATP is getting degraded, so minus one, and ADP is getting formed plus one, and one hydrogen H plus ion is also getting formed, which is plus one. So after this is done, you can uh, basically I'll read this out. So the process of generating a stoichiometric matrix S for a given reaction is demonstrated. Specifically, the conversion of 1 mole of fructose 6 phosphate to 1 mole of fructose 1,6 phosphate by the enzyme phosphofructokinase 1 PFK1 in the reaction V3. So the rows of S here, the rows of S describe the components of the network. This S vector. So this is only for this third reaction. You can construct a S vector S matrix for across all the reactions in your network. Now the rows of the S will describe the components of the network, while the columns will represent the underlying interactions, how they're getting formed or degraded. The stoichiometric uh, coefficients at the intersection of a row and column quantitatively captures the precise interaction with inputs delineated with negative coefficients and outputs delineated with positive coefficients. So basically, F6P fructose 6 phosphate is getting degraded and fructose di 1,6 phosphate is getting formed. So plus one for FDP and minus one for F6P. Now the C panel here, it basically describes the optimization framework, and here is the objective function that is chosen here. So objective function is you're maximizing your V12, so maximizing your the rate for the 12th reaction, and this is the objective function here. And as we said, the exchange reactions are just to note one thing, that the exchange reactions are excluded from this rendering for simplicity. So the exchange reactions are not present here, just to keep that in mind. And the basically the fluxes is shown here. So after the prediction of the model, how the fluxes are, the values of the fluxes are varying across each of the reaction are shown here. So if you see, the flux is quite less here in the first four reactions, and much higher on the lower side of the reaction. So these are not like shaded accordingly to the weights of this flux, but yeah, in general the idea is flux here is low, flux here is high. And now your objective function was to maximize this V12 rate. So this is something you can expect. Now, just coming back to the basically how you're calculating the v vector. So this is the dc/dt that we talked about. In that case, it was dxi dt. So here c is representing the concentration of the substrates. So dc/dt will be 0 here, which is basically s into v equal to 0, which you sum up. This is basically the matrix multiplication sum equal to 0, and you'll have a lower and upper bound on each value of. So you have a lower and upper bound. You have a matrix multiplication equal to 0. So you basically need to find the null space of the matrix S. And you have a maximizing function, or basically the objective function, which is Z. So this is how a flux balance analysis works. I give you an overview about like how it works, but yeah, this is the general idea of it. There can be certain like variations and exceptions. We'll talk about it in further classes, but yeah, this is the general idea of flux balance analysis. Okay, moving on. Now we saw that there's something called this objective function. This objective function that you're trying to maximize. In this case, it was the velocity of the 12th reaction, or the reaction rate of the 12th reaction. But this the the objective function is very important for FPA, and selection of the correct objective function, the appropriate objective function is important to get good results. So the FPA model's objective is to alter the distribution of optimal fluxes within the metabolic network, influencing damage accumulation and lifespan. To quantify changes in replicative lifespan and generation times, cell lifespan using various objective functions in the FPA model was simulated. This is taken from a study. So they took objective functions which included maximal growth. So these are functions that are all like if you are using MATLAB, so in the Cobra toolbox, there are certain functions that are already defined. I'll show you one of them. So basically here the objective functions that they took are maximal growth, minimal glucose uptake, maximal and minimal ATP production, etc., etc. And they did up to two successive optimizations. And flexibility was allowed by permitting violations of optimal values by a factor of epsilon, exploring a range of epsilon values. Now, glucose uptake rates were solely determined by objectives and constraint, allowing for unconstrained uses. Now, glucose basically did not have any constraints. Now, the simulation results now indicate that the choice of the objective function significantly impacted replicative lifespan and generation times. Cells without maximal growth optimization showed limited growth, while some failing to divide or doing so infrequently with long generation times, emphasizing the importance of maximal growth in replicative aging. So if you are looking at replicative aging, maximizing the growth can be a very good optimization function. So depending on the scenario you're trying to model, your optimization function needs to be chosen. So basically, this is a panel that is showing the basically how the objective function is changing the results. So this A panel is basically for generation times, and B are for cell simulated with different objectives and this flexibility of these epsilon values. Uh, so here you can see maximum growth i- is being shown basically using parsimonious solutions. The maximum growth, the objective function of the maximum growth is showing the best parameter counts. So these are basically wild type parameter counts, number of parameters here. So maximum growth is showing the most number of parameters here. So uh, basically cells without this maximal growth optimization showed limited growth. So importance of uh, this slide is I put here just to show you, just to give you an example about how taking different objective functions uh, can mess with your results. So using the perfect optimization function for for your particular scenario is very important. One such example here, I'll just show you here. Now, yeah. So this is one such optimization function. So this is a model that is uh, you can go through this. So this is a model inside Cobra that is basically showing this is for E. coli. I think, yeah, yeah. This is for E. coli metabolism. So this is the network that they are modeling, and yeah, just a second. Huh. So the objective functions that they have chosen. This is basically the tutorial. The different objective functions that they have chosen, they have given here, and the biomass production they mathematically represented by this optimization function. So if you search for this, you'll see the function here. So this is a predefined biomass objective function with GM. So there are predefined functions inside certain software, certain toolboxes that you can use for these kind of like known models. So I just wanted to show you the some examples here. Now moving on from the objective function, uh, we'll move on to something called minimization of metabolic adjustment. This is a variation of FPA. So what it is doing and how it is different from FPA is minimization of metabolic adjustment, which is MOMA in short, is an algorithm for predicting metabolic flux distributions after a gene knockout. So remember we talked about certain perturbations here. So these perturbations that you, you are performing on the network. So that can be mutations in the network structure, or that can be environmental stimuli, gene knockouts, whatever. These perturbations, you can measure the impact of these perturbations on the network and how these perturbation is affecting the network using techniques like minimization of metabolic adjustment. So the whole idea of MOME is I think I have not mentioned here. So yeah, the whole idea of MOMA, minimization of metabolic adjustment, is that even if a certain element in the metabolic network is not there, or it is perturbed, or it is not working properly, the system will choose to adapt in such a manner that there is least adjustment, and there's least change from the original network. So the edges that were flowing from one metabolite to another, the reactions that the different metabolites are involved in, the the maximum part of that should be conserved. That is the whole idea of minimization of metabolic adjustment. This is very like evident from the name as well. So you're minimizing the adjustment of your metabolites even after certain perturbations to the network. Now, this assumption for optimality for a wild type organization is justifiable. Now, the same argument cannot be valid for genetically engineered knockouts that were not exposed to long-term evolutionary pressure. So, this concept that we are talking about, uh, uh, here I'll just talk after this point. So, in the basis of this method lays the assumption that after knockout, metabolic fluxes undergo minimal changes with respect to flux configuration of the wild type. The thing that I said, like how with minimal changes to the network, you can get back the original network again. And now this MOMA employs quadratic programming to identify a point in space which is closest to the wild type point based on the conjecture that the mutant remains initially as close as possible to the wild type optimum in terms of flux values. How it does that, we'll see. But the point is, one point that I want to like highlight here. Okay, the assumption for optimality for a wild type organism may be justifiable, and the same argument may not be valid for genetically engineered knockouts that were not exposed to long-term evolutionary pressure. This term is very important, that were not exposed to long-term evolutionary pressure. Now this this argument that the minimum changes occurring in the network to get back the original network. This argument is only valid when there the organism has not been carrying that knockout or that particular perturbation for generations, so that it has evolved, or it has adapted to that perturbation. That if that has happened, so there will be some other rewirings as well, so that cannot be explained by the model. So that the concept of adaptation of a particular mutation, a particular phenotype to the environment, depends on if the phenotype is helping you survive more, or helping you reproduce better. So based on that, if the mutation is good for the organism, then the mutation will carry on, and the changes will be adapted to the environment. Now, if that is the case, then this concept will not be applicable, because then the whole network will be rewired according to the new system that is more stable than the old system. Now, if that is not the case, if it has not been put in an evolutionary, long-term evolutionary pressure, if the mutant organism has not been put under that long-term evolutionary pressure, the cell will try to bypass the knockout or bypass that particular gene in a minimalist way possible. But that is not true when there are generations of evolution involved in the in, in the organism. Now, coming back here, to predict the metabolic phenotype, MOMA determines a flux vector V in the flux space of phi of a mutant with the smallest Euclidean distance. I hope you all know what Euclidean distance is, which is basically differences in the x coordinate square plus differences in the y coordinate square plus difference in the z coordinate square, and you take a root over. It's basically it's the simplest distance metric that is possible. So s- so if you assume your W is your wild type organism's flux vector, and your V is the flux vector determined by MOMA, then there will be then when you calculate, basically this is basically the Euclidean distance between Wi and Vi, and after you calculate, you are trying to minimize this distance. So you are trying to look for points for which this distance metric is the least. So minimizing what you are minimizing here, you are minimizing the distance between the solution set of your wild type organism and your perturbed organism, knockout or whatever. It is what you are trying to do. So, and minimizing d is equivalent to minimizing the square of d, because there's a square root, and if you square it, it's the same only in terms of minimization. And if you have constant terms, say for example wi, you can like they can be left out from the objective function, because those are constantly. If you take a difference, that will be noted. Like it will be zero basically. Okay. Now, this is how basically on the left side of the of the slide you can see this picture. So this is how uh, FPA and MOMA are different. So uh, this A panel is showing the underlying. So first I'll just give you an overview of the figure. So basically these red lines are showing the two objective functions. So the objective function that is satisfying the wild type animal, wild type network, and giving an optimal solution is this line. And this is a basically these are all the constraints. So this boundaries that you see, these are all your constraints, and this is your objective function. So your objective function is crossing your constraint at this point, so this is your optimal solution. This is the FPA of the wild type, which is the optimal. Now the FPA of the knockout, you get it from the objective function of the knockout sub basically knocked out network. This is basically the green space is showing the wild type, FP of wild type, and the yellow space is showing the physical space of the knockout network. Now, so yeah, it is saying the same here. A schematic 2D depiction of the feasible space of the wild type and for the mutant flux J is represented by the green and superimposed yellow polygons. The coordinates are two arbitrary representative fluxes, an extremely simplified version of the multidimensional flux space. Like you can imagine this to be like an n-dimensional space, but just for representation in here, it is shown as a 2D space. The solution to the FP problem is the point that maximizes the optimal solution. So this is your maximum optimal solution here. So you're maximizing your objective function. This is the point. And uh, the solution to the FPA problem is the point that maximizes the uh, objective function. An optimal FPA prediction can be computed both for the wild type and the knockout in a similar manner. So this is your highest FP of the highest, uh, basically the maximum optimal solution here. And this is so for MOMA, this is the highest point, and for wild type, this is the highest point according to the objective functions. Now, the alternative MOMA knockout solution C, there's another solution. So MOMA, you have seen this solution B, and there's another solution C. This is an alternative solution. The alternative MOMA knockout solution C, calculated through quadratic programming, can be thought of as a projection of the FPA optimum into the feasible space of the mutant. So if you imagine this and this, so the MOMA of the knockout will is kind of looking as a projection of the of the FPA of the wild type. Now, constraints, no, yeah, yeah, projection of the FPA optimum into the feasible space of the mutant. The mutant FPA optimum and the corresponding MOM solutions are in general distinct. So these are distinct. Now, robustness analysis for partial deletion has been done in this panel, and constraints corresponding to the partial gene knockout leads to a feasible space that is intermediate between the wild type and the mutant. On such intermediate spaces, both FPA optimum and the MOMA projection can be calculated. So, just to give you the overview of what it is trying to say here. So, if this is the optimum solution of given by the objective function on the knockout, and this is the FP of the wild type, the optimal solution. So, and this is another solution, MOMA for knockout, which is suboptimal. This is not the optimal solution because this is not passing through the through this objective function. Now you have to calculate the distances between these points between the MOMA of the knockout and the FP of the wild type, and you see which distance is the lowest. So uh, on such intermediate spaces, both FP optimum and the MOMA projection. So your this is your FP optimum, and this is your MOMA projection. So optimum, that's the projection. This is not the optimum for MOMA, just to keep that in mind. So the optimum and the MOMA projection can be calculated. A robustness analysis method is unique to MOMA. One can perform a sampling of the feasible space around the optimum found by simplex in FPA, point A, which is this point, and find the corresponding MOMA projections. The robustness of the MOMA solutions is calculated for the defined threshold of sensitivity of the objective function for endpoints obtained by limiting the corresponding fluxes to a given fraction of their wild type value. So basically, these should be a projections of these. Then you calculate the distances between these. So you can have, say, for example, this is your objective function. It is having three such. Yeah, so A and A dash are on the basically the optimal space. A hash here, it's not in the optimal space. Now you can have projections of these onto the MMM space, and then you calculate what are the distance and you try to minimize the distance. The uh, basically the robustness of the MMM solutions is calculated for a defined threshold of sensitivity of the objective functions, and this method is useful also for studying possible multiplicity of MMM solutions in case alternative LPA solutions, optimal solutions are present. So just to give summarize the whole thing. So this is your wild type space. This is your mutant space. You have a optimal solution with the help of objective function. Then you project this onto the wild type space, a knockout space, and also in the knockout space you have a point which is the optimal solution according to the objective function. Now you check the difference distances between A and C and also A and B. Check which is greater. If A and C is smaller, that is a more possible that that is a more optimal solution than B. So what it is trying to do essentially is trying to minimize the distance between this boundary and this boundary. That is the whole idea. So to summarize, MOMA will calculate the Euclidean distance and will try to minimize it between the optimal solution of the wild type and not only the optimal solution, also the suboptimal solutions of the knockout. Basically, this is a triangle method. You will check which distance is the lowest and consider that accordingly. Okay. So this is another a mechanism for handling perturbations in a metabolic network. This is basically the short form is ROOM. It, the full form is regulatory on-off minimization. So traditional metabolic modeling techniques like MOMA, which we just saw, have been invaluable in predicting cellular responses to genetic perturbations. However, MOMA's approach, while effective, has limitations in capturing the full scope of the regulatory dynamics within the cell. Now, how is this ROM room basically different from MOMA? So first key difference is the norm selection. So MOMA, as we discussed, minimizes the Euclidean distance between flux distribution of the wild type and the perturb strain, while Room employs a different norm. Room focuses on minimizing the total number of significant flux changes, emphasizing the cell's adaptive cost reduction. We'll see through these three examples in the next class how these are actually done in real life. But yeah, this is the general idea. Also, Room incorporates regulatory dynamics as well. It acknowledges that genetic regulatory changes post gene knockouts are minimized by cell surface to reduce the adaptation cost. So uh, basically these regulatory constraints that we talked about, these dynamics and the constraints are okay. I'll talk about it after I finish this. So it operates under the assumption that the regulatory changes can be parsimoniously described by Boolean on of dynamics, assigning fixed cost to each regulatory change irrespective of its magnitude. Also, there are regulatory constraints as we discussed in the beginning. So MOMA and also FPA, they do not consider this regulatory constraints, these transcription factors that can regulate the gene expression. There are other factors that actually regulate the gene expression. So unlike MOMA, which does not explicitly incorporate regulatory constraints, Room implicitly accounts for regulatory changes by identifying significant flux changes in metabolic reactions. This is essential as efficient expression likely requires adjustments in flux implicating changes in gene expression levels. And also, uh, Room has a preference for growth solutions, while both MOMA and Room aim to find flux distributions close to the wild type. Room's matrix implicitly prefers high growth rate solutions. So it will always try to maximize the growth rate. So this preference leads to more accurate predictions of post-adaptation states as evidenced by its ability to minimize significant flux changes while maintaining the situation. We'll look into the examples late in the next classes, so it will be more clear then. But this is just to give you an overview. So this is a network. So just to compare Room and MOMA. So this is an example network. So these are your boundary conditions V1, V2, and V5. Now uh, it's this in the figure A, a given flux distribution for the wild type. This is for the wild type intact network, uh, uh, which is obtained by basically FP and experimental flux data. The flux through B2 represents growth rate. So this is representing growth rate. Now MOMS MOMS prediction for knocked out network following the knockout of V6. So this sixth uh, reaction. Just a second. Now the sixth reaction here, converting C from E, now this is deleted. So uh, after deletion, this is the MOMS prediction of this is basically just remember B2 is the growth rate. So B2 is basically 4.4, B3 is 4.4, and this is 8.8. Now this is MOMS prediction. Look at Room's prediction. This is the C is the Room's prediction. Room's prediction for knocked out network. Room finds tangents in flux only along a short alternative pathway through V5 and V4, preserving the optimal growth rate of the wild strain. And also notice it is maximizing this. This the B2 here. Basically, the growth rate is more here, more for room compared to MOMA. Now, yeah, yeah. So in all the figures, the metabolic branch point is this B point, along with the reactions that produce or consume it are highlighted. So this is in this model, our B was our point of interest. Uh, just note that in Room's prediction, linearity of flow is preserved in the branch point B as opposed to MOMS prediction. So just to like what to say, just to show you how differences are there in the prediction. I'm not going into the details of each of the reactions, but just to give you an overview how the results are different. So yeah, in Room's prediction, linearity of flow is preserved. Now note that also in the MOMA solution uh, has a Euclidean distance 62.8 from A, which is the Room solution, and all basically the yeah. So the MOMA solution will have a Euclidean distance of 62.8 from A, and Room solution has three altered fluxes with respect to A. On the other hand, MOM solution has nine altered fluxes with respect to A, and Room solution has Euclidean distance 75 from A. So, depending on what criteria you're trying to maximize or minimize, Room and your basically MOMA are giving different predictions, but like you need for your specific model you need to decide on what type of uh, perturbed analysis you are trying to do. So are you trying to maximize your uh, basically um, in this case the B2 here, so this is basically the growth rate. Are you trying to maximize the growth rate? If so, then the room is a better uh, prediction method. If you're trying to minimize the distance between the wild type and the perturbed or the knocked out organism, then MOMA is a better alternative. We'll look into examples in next classes, but yeah. So yeah, this is all we have for the lectures. Now we'll solve some MCQs. I'll do the same. I'll put this on the screen for 2 minutes. So not these questions for 2 minutes, maybe because these are easier. So these are possibly for 1 minute or 2 minutes, depending on the type of the question. And please send the answers in the chat box. And I'll be back in just a minute. I'll confirm the. Excuse me for a minute. Start doing this. Mm, okay. Thank you, Ritvik. That's true. So constraint-based modeling approaches are
Could you
always.
unmute?
Oh, sorry. Uh, Sorry. Yeah. So the answer is correct. So constraint-based modeling reduces the space of possible flux distributions in a metabolic network by imposing constraints on the network. So the idea is like if, if you have a set of solutions and your number of unknowns are basically more than the number of equations that you have a space of solutions. So and the space will be very big. If you want to constrain if you want to put some boundary conditions on those, then the space will be smaller after you put the constraints. And the whole idea is to reduce the space of possible flux distribution so that you can have uh, the, the least number of basically possible optimal solutions because your solutions will not be unique. So yeah, this is true. Next. No, okay. I think this is the same question. Yeah. So, we have the answers. 1C, 2B, 3A. Okay. So you're talking about option number two. Yeah, that is correct. So if your number of metabolites is greater than your number of reactions, so you'll have a tall matrix. So if you have a tall matrix, it's basically over-determined. So, and if you have a fat matrix, so number of reactions that are more than the number of metabolites, you'll have undetermined solutions. So, yeah, the second option is correct. So, 1C, 2B, 3A. So, 2 is basically just for your information, 2 will be a tall matrix, and 3 will be a fat matrix, and 1 is a like symmetric matrix. Yeah. Okay, next. That is the correct answer. Stoichiometric constraints. And don't think there is much to explain here. Moving on to the next one. Two, three. Yeah, the value of R is unique, and for different Vmax, we can get. Sorry, oh, what?
No, no, I said the wrong information. I said for
No,
different VMAX they get different values of.
yeah. Okay, so you said two and three, na? Right.
Yeah, this is one and two.
Okay. The maximum flux of these reactions are. Okay, like the value of the flux is not
Yeah, I got it.
determined by the value of Vmax.
Uh,
So like
for a
different
moment,
Vmax,
yeah.
the value is
No,
not
we are
constant.
changing the bounds, but essentially this remains the same.
Yeah, yeah. And your value of R must be unique. So
Yeah.
okay. One, two, three. One, two, three. Why not four? Like,
For
what's the
four, like I was sort of confused with this term, like with this constraint of like ensuring survival, I was sort of considering if
So survival, you can think of a regulatory constraint that you're
constraint. Okay.
putting on the, like maybe for example for one gene, that if that gene is mutated, uh, you get a basically a null mutant. So basically your cell will die, or it's lethal mutant or something like that.
Yeah, yeah, yeah.
And you are putting a regulatory restriction on that gene will not be activated, or that gene will not be, if it is repressed and the cell is killed, then that gene will not be repressed, or that gene will be formed, or whatever the function of the gene is. So you can think of it as a regulatory constraint that you're putting on top of like this mass
Yeah,
action and energy
yeah,
balance.
yeah.
Yeah.
Okay, okay.
So all the four options are correct.
Yeah, true.
Yeah, it's true. It's nothing much to describe here. So yeah, so MOMA will calculate the Euclidean distance between your optimal point in your wild type and your and the and the projections and also the optimal points in the knockout or the perturbed solution set, and it will find Euclidean distance between those points and try to minimize the Euclidean distance and find the corresponding points with the minimum Euclidean distance. That's what it will do. So yeah, it is basically trying to minimize the difference between the flux distribution after perturbation and the wild type flux. Let's see. 2 and 3. Okay. No, I know. Yeah. So yeah, identifying steady state flux distribution in a metabolic network, that is a prime example of FPA, usage of FPA. And yeah, it's based on linear programming, so it is we're trying to minimize or maximize the objective function using LP. This is not true. This may seem to be true. An unconstrained technique to determine flux through every reaction in a metabolic network and identify the phenotype, but we cannot identify the phenotype using FPA. So it's just the uh, the solution set you're getting, the set of genotypes or the set of uh, solutions that are possible. So phenotype and getting from flux, you are not getting from flux balance analysis. So moving on.
Is it okay if I just say the answers out loud?
Yeah, yeah, because no one else is there. I think yeah, someone is there, but they're not asking. Sorry, answering. So yeah,
So yeah, I think this the second one where any reaction can be an objective function, and the fourth one, yeah,
yeah, that's true. Just to keep this in mind, reaction chosen as an objective function need not be balanced. This is not true. So
yeah,
balanced as in the stoichiometric balance. So
yeah,
your reaction should be stoichiometrically balanced so that you can have the proper stoichiometric matrix. So
yeah,
that
yeah.
is important to have.
Yeah.
So yeah, this that can be a tricky answer, but yeah.
Mm.
Okay, moving on. So this is the last question. These are like three calculations. Just do it and put it in the chat, or like if you want to tell it.
Sure.
Yeah. So these are like three elements of the vector, 1, 4, and 2. This is not 142, if just to be clear. Yeah. You don't have to answer till 4 decimal places. 2 is enough. Like if you can do 4, otherwise 2 is enough.
So for the first one, it will be square root of 22, which is some 4.69 something.
Okay. Second one.
Yes. Go to second and third and then come back.
Oh yeah, yeah, yeah. Take
Yeah.
your time. Take your time. Give the three answers together.
So for L1 norm,
Hmm.
uh, it will be 8, and for infinite norm, it's 3.
Yeah, it's correct. So I'll just repeat it
Mm.
for everyone. So yeah, V1 and V2 you have two vectors. you're finding the basically the Euclidean distance. So uh, just there's one error here. So this should
Brackets.
be in brackets. Yeah, the bracket is not there. So anyway, the calculation is correct. So you get root over 22 here. So 4.69. For L1 norm, it's just the absolute differences between the coordinates. So x
Mm.
minus x1
Yeah.
minus x2, x y1 minus y2, y
Mm.
z1 minus z2.
Mm.
Just the absolute differences. You add them up,
Yeah.
you get 8. And for infinity norm, it's the maximum of those differences, absolute differences. So maximum of 1 minus 3, 4 minus 1, and 2 minus 5.
Mm.
So it's 2, 3, 3. The answer is 3. So yeah, that was all for today. If you have any questions, you can ask, or we'll drop off. So like, I just want a overview of like, I are these sessions going fine, or do you want me to change anything? Tell
Yeah.
me.
No, for me personally, like I think we have discussed this before. The same thing where, like, for me, this system works really fine. Where, like, after listening to the classes, where been introduced to a lot of courses, and after reading supplementary material and everything like that, I have an idea of what the subject matter is. But then these sessions essentially reiterate those concepts, so
Mm-hmm.
I get an idea of, okay, what did I miss, or like there are some finer details which I missed or not, which sort of gets and the questions usually help because there are some concepts where I know them theoretically, but then I only get to sort of understand them better when actually doing a problem or answering a question or something like that. With this and the assignments and things like that. So like for me, this works really well.
Okay. So the questions are essentially like these are the question types of questions you can get in assignments. So
Yeah,
the
yeah,
questions are basically like similar to what you'll get in
yeah,
assignments.
yeah.
And regarding the content, I try to like I watch the original videos and I try to make the slides a little bit different, a little bit more
Mm.
intuitive, like not so much mathematical, a little bit more on the example side,
Yeah,
how
yeah.
the biology is working on. I try to do that, but
Yeah.
yeah, if you want if you want, it can be I can make it more mathematical as well. So if you want that, let me know. But yeah, other than that, if you have any thing to mention regarding the content of the, you've read the whole content on the
No.
on the original videos, and
Mhm.
also like you're seeing the content here as well. So
Yeah.
if you want to have a little bit different content as to compared to the videos, or a different style of content that I am doing here, so you can let me know about
Yeah.
that as well. Any changes you might want.
For now, at least for now, like I don't know, like I mean, it
It's okay. It's
seems to
okay.
work fine.
There's no
Yeah, yeah.
pressure.
No, it seems to work fine.
Okay.
Just
Okay
thinking
then.
if like
I just want to tell you, just let me know whenever you feel that this
yeah,
is
yeah,
not
yeah,
going
sure,
well,
sure,
or
sure, sure.
because this is my first time as well taking
Yeah,
notes,
yeah,
so yeah, I have a lot to learn as well.
yeah,
So yeah. Okay. Then we'll stop it here.
yeah,
So yeah, thank you for
yeah.
joining.
Thank you,
We'll
thank
see
you. Yeah,
you next week. Thank you.
sure.